Okay, so our final video on cellular respiration, we have glycolysis, which we've talked about. We talked about the generation of pyruvate and how we have two pathways which we can follow at this point: a pathway where oxygen is present and a pathway where there is no oxygen present, and which leads to fermentation. If oxygen is present, it enters the citric acid cycle, or the tricarboxylic acid cycle, or the Krebs cycle, this cycle over here. And we've generated so far 10 NADH, 2 FADH2, 4 ATP, and we've lost 6 carbons as CO2. So we've really lost all of our carbons for glucose. It's been fully stripped down at this point. But the next step is our biggest return on investment, and it's going to use primarily these molecules, NADH and FADH2. So let's take a look here. This is kind of a simplified version of what we're going to be looking at. Our citric acid cycle, of course, occurs in the mitochondrial matrix, which is shown here, these membranes. This outer membrane is the outer mitochondrial membrane, and you have your intermembrane space here. The next part we're going to be talking about are these little enzymes, 1 through 4, as well as this ATP synthase over here, which is going to generate a lot of energy for us, as we're going to find out. I would like you to note where it takes place. Again, your cycle, we know that takes place in the mitochondrial matrix. It's generating a lot of NADH. A lot of NADH is being generated in glycolysis and some in the link reaction as well. But this is where NADH is, there's tons of it being generated. Very important. So let's take a look at this electron transport chain more in detail. And this is generally how it works. You have NADH molecules that are kind of moving over here, and they're going to split with their H plus ion here. So you're going to end up with NAD plus, H plus, and you're going to end up with, if you notice that these are positively charged, see how those are positively charged, you're going to end up with 2 electrons. So you end up with 2 electrons and an H plus, and what's going to happen is those electrons are going to be passed through these proteins. And as they're passed, there's little transfers of energy that occur at each of these steps as this electron gets passed up through this transport chain. And that energy is used to pump these H plus ions that the NADH is bringing over here. It's used to pump them across this membrane into the intermembrane space. I'll put this up here, intermembrane space, just so we remember where these things are working. And this is the matrix, mitochondrial matrix over here. Okay, so H plus ions are being pumped up here through all of these proteins, these 1, 3, and 4. You don't need to know the name of these, NADH reductase. It's just interesting to know. Cytochrome C is a very crucial molecule to this. In fact, you'll see possibly a question on the AP Bio test about comparing cytochrome C DNA analysis across species. This is where that molecule is found, and the reason we like to compare this one is because it's so highly conserved, because these proteins in here are so crucial to cellular respiration that they can't withstand a lot of change. So FADH2, this is going to turn into FADH plus 2H plus 2 electrons as well. So you've bought a bunch of electrons over here. You're using that energy for pumping them through these proteins over here, through this membrane. As it's changing hands, it's dropping off little bits of energy in each spot. As it changes, there's energy being used in every little spot here to help pump these H plus ions. Well, as you are probably going to notice, you're going to end up with a bunch of H plus ions up here. You're going to form what you will remember as a concentration gradient. So, which way are these H plus ions going to want to travel? If there's a bunch of them out here, well, they're going to want to travel through this membrane. Unfortunately, there's only one way through, and that's through this very special molecule called ATP synthase. There's a very thin channel in this ATP synthase in which these H plus ions are going to kind of flow down. Of course, if you've used any of the sinks in my room, you'll know that if you try to force a large amount of something through a small conduit, it's going to generate a lot of pressure. And so this thing generates a lot of energy by shooting through this ATP synthase. And as these H plus ions shoot through here, this ATP synthase molecule kind of rotates, and it goes really fast. It's kind of like it's analogous to like a windmill or something like that, the way it spins. And it constantly spins, and it slaps these phosphates onto these ADP molecules, and generating ATP in very high quantities, over 30 ATP from one glucose molecule, which is a lot. It's a lot compared to these other steps, right? This is what makes mitochondria so special and so important. Without having engulfed these mitochondria at some point in our past, we never would have evolved even to be multicellular organisms because we just wouldn't have the available energy. We wouldn't have been able to evolve into such complex multicellular organisms with muscles and things like that without the ability to generate massive amounts of ATP. And our mitochondria kind of give that to us. So this process here of the electron transport chain plus the chemiosmosis, which is a name for these H plus ions kind of moving down this concentration gradient through this enzyme here, ATP synthase, is the process of oxidative phosphorylation. Now, the only enzyme I really want you to know from this process is this one right here, ATP synthase, just because it's so crucial, such a crucial enzyme to this process. So let's go ahead and review what we have so far. Hopefully this picture will help you kind of put it all together, the big picture-wise. Glycolysis, 1 6-carbon glucose molecule yields 2 3-carbon pyruvate molecules, and you get 2 net ATP and 2 NADH. Link reaction, 2 carbons are lost as CO2, and 2 NADH are formed. Krebs cycle, you've got 6 more NADH and 2 FADH2, along with 2 ATP, and you've lost 4 carbons as CO2. So we've lost all our carbons that we breathe out. 4 plus 2 is going to give you 6, right? We've lost all of our carbons from that sugar at this point, and that's what we would breathe out, right? NADH and FADH2, these are the super important molecules that are going to travel to this next step and get us the big, big payoff in oxidative phosphorylation. So oxidative phosphorylation, NADH and FADH2 are converted to NAD plus and FAD, and approximately 32 ATP are generated. Oxygen is the final electron acceptor. Water is formed in this process. So you'll notice, let me go back to this image very quickly. You'll notice this character, I didn't discuss this yet, but we need oxygen for cellular respiration, right? But we haven't used oxygen so far in this process. Well, this is where it's used. This is where it's used. You have this O2 molecule binding with these H plus ions, also further lowering the concentration inside the matrix here of H plus, and it's accepting these electrons and bonding with the hydrogen to form water. So O2 is your final electron acceptor. I think that's another important thing to take from this lesson, that oxygen is the final electron acceptor. Water is formed in that process. The reason this is so important, as we're going to see, is photosynthesis is kind of the reverse in this process. When we start talking about photosynthesis, I'd like you to compare it to cellular respiration. I might review some of these slides during photosynthesis so we can see how this whole process is kind of flipped on its head and reversed. But yes, oxygen is your final electron acceptor, and water is formed in the process of cellular respiration during this oxidative phosphorylation step. Okay, I think we're good so far. Again, we've got a total of approximately 36 ATP. Again, this number varies depending on the textbook you're looking at, 30 to 40, somewhere in there. Approximately, though, 36 ATP are generated from 1 molecule of glucose. That's a lot more than we got just for glycolysis alone, which would have only been 2 net ATP, right? So, aerobic respiration, very important. And I think I'm going to conclude this video. I hope that helped.